Hello everyone, thanks for watching this short video. My name is Robert Valeris and I am a PhD student at the College of Veterinary Medicine at the University of Minnesota. I would like to show you an overview of this study titled Network Analysis of Swine Respiratory Microbiome. My co-advisor's research group studies Mycoplasma pneumoniae, a major respiratory pathogen in pigs. In their latest animal experiment, they collected 6 different samples from the respiratory tract of 30 pigs. One of those pigs was naturally infected with Mycoplasma pneumoniae and co-housed with 29 naive pigs for 8 weeks to study natural transmission. Their secondary objective was to study the respiratory microbiome. The samples were collected throughout the respiratory tract, from the nose to the bronchi. DNA from all samples were extracted and used for 16S ribosomal RNA sequencing at the University of Minnesota Genomic Center. The objective of this study was to explore the network structure of the microbial communities in the respiratory tract of pigs. The analysis workflow resembles a network. The point was either a count matrix or the corresponding relative abundance matrix. These matrices were used to build networks through 4 methods: distance-based networks, Pearson correlation-based networks, SPARS-CC, and Speakeasy. Both SPARS-CC and Speakeasy have been developed for compositional data, such as the microbiome, and Speakeasy makes use also of graphical lasso to achieve greater sparsity of the edges. After all networks were built, I cleaned them to remove all nodes that were not connected, and I calculated some centrality measures, specifically degree, betweenness, and closeness. Finally, I applied 2 methods for cluster detection: walk trap and multi-level optimization of modularity. These are the networks from the 4 evaluating methods applied to the nasal samples. You can appreciate that the 4 networks have very different topology. As a recurring theme, you will observe that Speakeasy outputs the sparsest networks, while SPARS-CC and especially the correlation-based networks are the most complex. Here are the networks for the oropharyngeal samples, the laryngeal samples, the tracheal samples, all of them with very different topologies. Networks from the bronchial samples are the sparsest, no matter what method is used for building the network. This finding agrees with the current perception that the microbiome in the lungs is composed of fewer bacteria than in other parts of the respiratory tract. Into the clustering results, I am showing you here the clusters detected by the walk trap algorithm in the Speakeasy networks from all the sampling sites. I will explore these clusters in more detail to find bacterial genera that behave as keystone members, which is the equivalent of a hub in other networks. In conclusion, the methods for network generation have a huge impact on the resulting topology, especially on the number of edges. Therefore, it is advisable to explore several methods instead of only one. Also, there is a lot of heterogeneity among the different sampling sites as for microbial network topology, and further characterization of these networks to identify potential keystone genera is needed. Finally, I would like to thank my advisors, my collaborators, and the funding agencies, and thank you for watching this short video.